Can you hear me? Can everyone hear me much better? All right. Ms. Seubert, I'm sorry. Can you explain low copy number DNA?
Sure. So low copy number DNA is a type of DNA process where going into performing the DNA testing on it, the individual knows there's going to be a limited amount of DNA information there. And so there is a procedure that can be performed to enhance the DNA analysis process to make even more copies, which we haven't really talked about, in the PCR process. So it's actually adding on some of the molecular xeroxing during the polymerase chain reaction when you're actually making multiple copies of DNA to enhance the end result, the DNA profile. So going in, you know you're going to have a limited amount of DNA, and so you're actually enhancing your DNA analysis process to adjust for that to make more copies to generate a result. And with that result, there's certain evaluations that have to go into those results because of the nature of low copy number. Since you don't have a tremendous amount of cellular material at the beginning, there are some things that in the end that have to be evaluated in order to generate the reliable or to have a reliable result.
Okay. So when we say low copy number DNA, I know these two terms are used interchangeably at times, but that's incorrect, right?
That's incorrect.
Okay.
Right. Touch DNA can be samples where at our laboratory, if a firearm comes in, and you think about the nature of when you hold a firearm and you put your finger on the trigger, your hand on the grip, you're coming into contact with it, but not necessarily for maybe an extended period of time. So that would be an area that we would swab, knowing that that's where an individual's had contact. I would expect perhaps to get a result for the deposit of skin cells, but I'm not going to enhance my technique to make more of those copies from my normal analysis procedures. I already expect to have limited amounts, but at the current, when I was in the unit, we didn't have procedures to perform low copy number, which is enhancing the analysis techniques.
Now, I want to skip ahead a little bit to when you actually get the readouts of the DNA results. Can you touch briefly on that? Because I don't want to get too technical.
So what the end readout or the profile is.
Correct.
Okay. Can I back up even a little bit more just to talk about the DNA process?
Sure.
Okay. Since we haven't talked about it, the actual DNA process, how that occurs is, let's talk about a swab. We've talked about doing a swabbing of the carpet out of the trunk, the spare tire cover. So that swab is put into a tube, and there are chemicals that are added because in order to do DNA typing, we have to get to the nucleus of the cell where all the DNA is housed. So those cells have to be broken open in order to really get to that DNA and unwrap it from where it's contained within the nucleus. And so we're able to do that by adding chemicals, and then that DNA is now taken through the process. And the next step after we're able to extract that DNA away from the item that it's contained on is what's called the polymerase chain reaction, the PCR. And what that is essentially a molecular xeroxing. It's like going to a copy machine and making billions of copies of that one page that you have that you need copies of, but we're actually doing that at multiple locations on the DNA. We're actually looking at 13 locations on your DNA, and so we're taking all of those 13 locations and making multiple copies of the DNA that's present. And so the next step then is to actually utilizing an instrument, the DNA is put into a capillary column through a process called capillary electrophoresis. And this instrument has a very small capillary column where the DNA in that liquid form is injected into this capillary column, and we're able to detect it through a process using a laser. And we talked about those RFUs, or those relative fluorescent units. What that's telling me is it's signaling that there's DNA present. And it's able to send that signal to a computer, which is collecting all of that information, and then it's interpreted into a DNA profile. Much like if you've been to the doctor and you have a heart monitor set up and you get an EKG done, you see those peaks on the EKG machine. That's what essentially my end product looks like, those peaks. And those are representative of the DNA profile from that initial test tube where we extracted the DNA out and we were able to remove it from the inside the nucleus of the cell, and now I'm able to see the end result and generated a DNA profile, which I now will use in my interpretation.
Okay. And what you get through your readout, which are called electropherograms, correct?
That's correct.
Okay. And what you get are some squiggly lines and some peaks, do you not? Among other things.
Yes, the peaks are what I'm most interested in.
Right. Okay. And what you talked about to the ladies and gentlemen of the jury are the 13 different locations of where you search for DNA, correct?
Correct, the 13 locations, yes.
And these are also identified as markers.
Correct.
Okay, points of interest, so to speak.
Correct.
Okay. And depending on how much DNA there is there, well, the peaks also indicate to you how much DNA is present at that specific marker, correct?
Correct. The RFU units will be higher depending upon the amount of signal that's being sent to tell the instrument that it's detecting the DNA that's passing by that laser and creating a fluorescence, which is being collected and is outputted into a peak.
Okay. So if you have, what are the cutoff levels for your laboratory when reporting out something as DNA being at that specific location?
To actually report a DNA type, the threshold for my laboratory at the time I was in the unit was at 200 RFUs. And that was our match interpretation threshold. So anything above 200 I could use to make a match with.
Okay. And there are other laboratories and other kits that can actually report lower levels, correct? That's just that 200 for you to report.
Laura, she can ask the question.
For you, that 200 level number, it's a determination made by your laboratory.
Right. That number was based on the validation by the FBI laboratory at the time when we were validating the instrument and the kit to do the DNA typing in the unit.
But it is common in other laboratories that sometimes the threshold can be lower. Is that correct?
Laura.
Other laboratories will have different interpretation thresholds. That is correct.
Okay. And can you explain to the ladies and gentlemen of the jury as to what your threshold is to exclude an individual?
Any information that falls below that 200 RFU interpretation threshold can be used for exclusionary purposes. But there is one more threshold that's not depicted on your chart.
Okay, and what is that?
That is at 50 RFUs.
Okay, and what does your threshold with 50 RFUs mean?
50 RFUs is the threshold for where a peak will be labeled a peak. That is our peak amplitude threshold, which is based upon the validation of where a peak will begin to be detected in the FBI laboratory.
Okay. Basically, some people will call these other items down here noise or artifacts.
There are other laboratories that may in fact use the threshold below that 50.
Okay. And if in your laboratory, if you see something that's 50, you can call it a peak.
That's correct. That is when the threshold for identify or labeling a peak begins.
And would you consider the laboratory thresholds that the FBI uses as conservative?
The 200 RFU threshold it's a relatively conservative threshold, yes.
Okay. And the 50?
Conservative? Well, it's based on the validation, so I would say that is a scientific threshold based upon the validation for when reliable results are generated.
Okay. Now, you told the ladies and gentlemen of the jury about you being able to call something at the level of 50 a peak, correct?
Correct.
Now, and that would give you an indication that there's something there, correct?
At 50?
Yes, at 50.
Correct.
But unless it reaches 200, you can't report it out.
If it is at the STR loci, cannot be used for inclusion unless it is above 200. There's an exception with the amylogenin or the sex typing locus where information can be used if it's below 200.